In this lesson, we'll be discussing the streak plate isolation technique that's commonly used in microbiology labs and colony morphology. The streak plate technique is a method of isolation. So typically when we collect a sample of bacteria and inoculate it to a nutrient agar plate, if there's a high concentration of bacteria, they do tend to just grow into a large blob. To kind of show you that, it would be something like we'd expect to see here. And the problem with something like this is there could be unknown contamination hiding within the bacterial growth that we're trying to generate, which could obviously affect our test results. So typically when we want to inoculate a plate to produce bacteria for further testing, whether we want to do staining or selective or differential testing, we want to inoculate the plate in a way that we get these nice isolated sections that we can collect samples from. So in order to do that, we do a streak plate technique, which is basically a dilution technique where we dilute out the bacterial sample over a series of 4 quadrants so that by the time we get to the 3rd and 4th quadrant, we have increased our chances of getting these isolated bacterial growths. So typically how it begins is we collect a sample of bacteria and we pass it through what we call quadrant 1. And then we actually sterilize our inoculation loop. And then after we've sterilized our loop, we pull a small sample from quadrant 1 downward so that we can make quadrant 2. Then again, this is very repetitive. We sterilize our loop, and then we take a small sample from quadrant 2 and pull it down to make quadrant 3. And then for the last time, we sterilize our loop and we take a small pass over quadrant 3 and pull to make quadrant 4. And what you're doing is you're putting less and less bacteria in each quadrant so that those bacteria have the space to grow in isolated regions and mature into the structures that we call colonies. So once we incubate the streak plate, we fully expect that typically quadrants 1 and 2, because they still have a high concentration of bacteria, we don't expect that they're going to really have isolated colonies. We expect that those quadrants have so much bacterial growth that the colonies kind of merge together in that big blob I was kind of referring to. But typically by the time you get to quadrants 3 and certainly quadrant 4, you have significantly less bacteria where they were able to be placed in separate regions where they could mature into these isolated colonies. And then from that isolated colony, which is each of these little circles here, you could collect a sample that you could feel confident about in regards to not being contaminated. You could collect a sample and put it on a microscope slide or run selective and differential testing on it. So here's a better look at what a real streak plate would look like. So the streak plate was very well done. We again fully expect that quadrants 1 and 2 have a very high concentration of bacterial growth. So those colonies merge together to see these growth patterns that we're seeing here. But by the time we get to quadrants 3, and again definitely in quadrant 4, we have these nice isolated colonies that we can use to pull samples from. But again, a colony really is just where one bacterial cell was placed, and then over a 24 to 48 hour period, that bacterial cell continued to grow and divide. So 1 became 2, and 2 became 4, and 4 became 8, etc., until there was thousands and thousands of bacterial cells. And at that point, it forms this macroscopic, kind of large visible cluster that we can see and we know as a colony. But it's important to note, this colony here doesn't just have one bacterial cell in it. It has thousands and thousands and thousands of bacterial cells, but they all originated from one cell that was placed there 24 to 48 hours ago. But since they all originated from that one cell, they have the same genetic makeup. It's a pure culture, meaning it's the only species within that colony. So when we collect a sample from that, that's how we can be assured that we don't have to run the risk of things like contamination when we're doing other tests later on. So kind of step one of being a microbiologist is be able to master the streak plate technique. Because in order to do all the other tests that you do in microbiology, the stains and the media test, you have to have a bacterial sample. And to get a bacterial sample, you have to have a nice isolated colony to pull from. And to get a nice isolated colony, you have to master the streak plate technique. So this is just a general overview again of the procedure. It basically starts off with sterilizing your transfer tool, which in this case is a loop that's being sterilized over a Bunsen burner flame. Could also be sterilized in an incinerator. Then you collect a sample of your bacteria, and you go in and you zigzag to make quadrant 1. Once you've made quadrant 1, you again sterilize your loop, go in, pull down a small sample from quadrant 1 to make quadrant 2. Then again, sterilize your loop, pull a small sample from quadrant 2 over to make quadrant 3. Again, sterilize your loop, pull a small sample from quadrant 3 up to make quadrant 4, sterilize your loop. And then at that point, you could set your loop down, you could label your petri dish, and then you could parafilm it and then put it in the incubator for about 24 hours. And then hopefully when you get it out of the incubator, you see a nice perfect streak plate that resembles this one with Serratia marcescens, which is a pink bacteria growing on it. So this is kind of ideal. These ones here are plates where the technique went wrong. So to kind of look at these, in this one, it looks like a streak plate technique wasn't even performed. It looks like whoever inoculated this one just took their bacterial sample and zigzagged it across the entire plate. So really failed to separate it into those 4 separate quadrants. And we also see that there's this colony here that looks different from all this other yellow growth. So this is likely contamination here. Then we have this streak plate here where we can see there was an attempt at doing the streak plate technique, but we don't have any isolated colonies. It's just kind of a solid growth where we did the streak lines. A couple of things probably happened here. One, possibly starting out with too much bacteria. Also, it looks like it was an attempt to fail to incinerate the loop in between each pass. So typically, like I said, you do streak 1, and then you sterilize your loop to get the extra bacteria off. And then you pull a little bit from quadrant 1 into quadrant 2, and then again you sterilize your loop and then pull from 2 to 3, and then again you sterilize your loop and pull from 3 to 4. What looks like possibly happened here is that the loop was not sterilized in between to get the extra bacteria off. So they did quadrant 1 and then just immediately pulled 1 to 2, 2 to 3, and then 3 to 4. And if you don't get the extra bacteria off, all you're really doing is stirring your sample around. You're really not diluting it like you're supposed to be. It also looks here like they may have pulled too much bacteria from each quadrant over. Typically, when we pull from the previous quadrant, we like to only go through a small section and maybe pass just once or twice. And it looks like in several of these, they went over 3 times and through a large portion of the prior quadrant. This one's not a bad streak plate by any means. I definitely do see some isolated colonies in there, so I would consider it a good one. The main concern here is this yellow growth. This yellow growth is unlike this kind of white circular growth that we see here, so this is likely some contamination here, possibly from not using the lid as a shield, like maybe the lid was opened up all the way, or maybe there was some contaminants on the inoculating loop that was used. So once we have those nice isolated colonies, we can actually use them to help us identify what type of bacteria we're looking at. It's not definitive. We definitely need to do more testing to confirm the bacteria, but it does give us kind of an idea of what we're looking at. So we need to talk about colony morphology, which is basically different characteristics that each bacterial species have that relates to the appearance of their colony. So for example, what size are their colonies that they typically produce, what color, what shape, are they flat, are they elevated, what do their outer edges look like. So for example, Micrococcus luteus is a bacteria that typically produces these nice yellow, perfectly round colonies, whereas something like Staphylococcus epidermidis would typically have round ones. Escherichia coli or E. coli, they typically have kind of these lighter greenish tannish colonies that are more irregular in shape. Serratia marcescens is typically these round pink colonies. So different species of bacteria have different types of colony morphology. So you'll learn to look at the colonies and kind of describe them based on their colony form, which is shape, whether they're really, really small, which is punctiform, or perfectly circular or irregular. We also look at things like elevation, whether they're flat, raised. Sometimes they're raised higher in the middle, which is umbenot, or even have an indentation in the middle, which is craterform. We also look at their outer edge, whether it's nice and smooth, which we call entire, or if they have, for example, large protrusions of lobes, which we call lobate. So you can use this image here to kind of look at colonies and describe them based on the characteristics here. There are other things that we look at as well. So for example, color is one factor. Serratia marcescens typically grows this pinkish reddish color, whereas Micrococcus luteus typically grows yellow. Most types of Staphylococcus are kind of like a whitish tan. So most bacteria kind of have their characteristic color that they grow. We also look at texture. This does imply that you're touching something, but you're not. So if the texture, if you look at the colony and it looks very kind of matte and not shiny, we typically say it has a dry texture. Then there's mucoid and moist. Mucoid has more of like a thick, like syrupy consistency, whereas moist is more water-based. Then we also look at basically how light passes through the colony. So if there's pretty much no light passing through the colony, we say that's very opaque. Then there's translucent, which allows a certain degree of light to pass through, and then transparent, in which all the light is passing through. This image shows you the various different types of colonies that bacteria can form, so I think it's good to kind of look and kind of try to identify them based on the terms used in the prior chart. So for example, here we have these very small punctiform colonies that are yellow, round, and opaque in color. Down here we have some orange colonies that again are round and have that entire or smooth margin with an opaque color. We also have some here that are brown and have an irregular shape versus like a perfectly round shape and have more of a translucent coloring. So it is helpful just to kind of look at these different colonies and identify some of those colony morphology terms.